this video, we're going to talk about this template param.txt file that specifies, well, it's a file that could specify all the parameters that MetaPathways needs in order to do the pipeline. First thing is off, any line with this hash character at the front is ignored, and so they're just comments. And you can write little messages to yourself about particular parameters, and we've sort of tried to help out by giving a few notes here. So the first setting here is this input format, and so you can either give the pipeline a FASTA file or a GenBank file, and if it's a FASTA file, it is of course unannotated and it will search against the sequence databases. But if you give it, for instance, a GenBank that's annotated, it'll take the annotations out of there and forego doing any protein blasting. And so that's a quick way to make a PGDB just from a GenBank file. And then if you just want to extract the sequences out of your GenBank, you have a GVK-unannotated, and that will pull the sequences out. And then basically you're treating a GenBank file, you're just using the GenBank file for the sequences. And the next step, there's a quick quality control, and basically we just have two parameters. It's that you have a minimum length that you need your sequences, so sometimes you don't want very many small reads to go through in the analysis, so in this case we have 180 nucleotides, and so that's just got to be a number. And then delete replicates, and so this is if you have a duplicate sequence in your FASTA file, it will just take one of them. Next step is ORF prediction parameters, and so one is specifying the algorithm. Right now we only use prodigal. That might change in the future. And then also there's a sort of a minimum length that you want your open reading frames to be. And perhaps you're not interested in small proteins because you don't trust them or something. Now the next part is you want to annotate those ORFs. And we have an algorithm setting here. You can do BLAST, or you can use the more efficient LAST to do it. And then you have this famous line here, which specifies which target protein databases you want to search against. And right now, we've actually only included two for you. So Metapsych v5, 11, 10, 21. Is this file right here. And so these names have to exactly match. If there's any, even if it's off by a little bit, it won't pick it up properly. And what it does is it looks for FASTA files that have that name in your BLAST database folder, and then it will try to format the database and do that. It'll do a whole bunch of all the sort of nasty BLAST stuff on the fly for you, but you have to get the names nailed down right here. And so the other one is the clusters of orthologous genes database, and both of those, well, the Metapsych one's getting a little old, but the COG is fairly recent. And of course you can add others, like mainly the main one missing from here is the RefSeq, but that's a really gigantic database that you need to obtain from the NCBI. I'll go through that in another video. Next, we have sort of annotation settings on this. So we have a min BSR, which stands for bit score ratio, and so it's a bit score of you and the target versus you and yourself, and so that's a ratio. And so generally having a bit score of better than 40% is a good idea. This has to be a number between 0 and 1. And then a max E value, we found that 10 to the minus 6 here is a good E value. Min score is the minimum bit score that you want out of your annotations. A length score is the minimum length that matches. And then the next is the maximum number of hits you want to see per query. And usually a number between 5 and 10, or maybe 20 at the most, is usually pretty sufficient. Unless, of course, you have other specific goals in mind, and in which case these settings are not for you. But generally for our metagenomes, these have been pretty good. All right, the next one is going to be a nucleotide blast from the QSEED nucleotide sequences. And so this one, it's same as above, except you have you specify here now nucleotide databases, and I've only included, well, we've included one, but then we also downloaded one. So I simply, if I need to add this guy to the list, I just copy the name from the BlastDB folder and then add it succinctly like that. Save this file. E-value, identity, and bit score are the main thresholds that we use. Next, there's pathway tool settings, and well, there's only one, and it's basically whether you want to use taxonomic pruning or not. For metagenomes, you want that turned off. Maybe for single cells, if you're closer to a particular taxonomy and you know what that is, you might want to turn it on. That's like heavily untreaded waters. Nobody's actually done that yet, so you want to be the first, go ahead. Next, we have grid settings. So if you've configured passwordless login to your local compute grid, in Canada, this is the Compute Canada, under any grid under the Compute Canada umbrella, but these could be anything. Most universities have a local supercomputer. Well, most grids have certain rules when you submit jobs. So, for instance, in this case, the batch size would be the number of sequences that you want to blast per job. So this is sort of like the unit size of your blast. And the next one here is max concurrent batches. There's often rules about how many jobs that you can have at one given time when submitting to a grid. And so in our case, we say keep submitting until you have 400 jobs outstanding, and then wait. But then keep the grid, always keep 400 in the queue. Yeah, if you try to submit everything to a grid all at once, that'll just cause problems with the grid. So either you have to increase your batch size, but if you increase your batch size, you'll start using more memory, and then it's hard to get scheduled. And I'll talk about more about that later. So the wall time on the SunGrid engine is the maximum amount of time that your job can run before getting kicked. Because you have to, basically when you tell this thing, you have to specify how much time you expect your job to run in, and you have to specify how much RAM or memory. So those are two parameters. However, when you do specify them, it basically affects, depending on how the queuing schedule works, this could affect how many jobs are done and when they get scheduled, depending on how busy the server is. So yeah, you have to play around with the batch size, the concurrent batches, and wall time and RAM, and figure out what's good for your system. So these settings turn out to be fairly good for our systems. So we've put them in there as default. Next up, you have your username and, of course, where the grid sits. So this is the SSH username that you use to connect to the grid. And then you have this, of course, the address of the grid itself. So this could be like an IP number, but generally it's, I don't know, my grid at some kind of address.com or .ca, because this is Canada. Next, you sort of define which steps of the pipelines you want to run, and some of them you can skip and others you can't. There's three sort of settings for each. One is a yes, you can skip it, you can redo it, or you can stop. And for each one of these, it's like preprocess your FASTA, annotate ORFs, change those things to amino acids, filter them, compute ref scores before going into the blast. So this is a self-blast done locally. Blast refdb. And so blast refdb is the only one that you can set to grid currently. And if you set that to grid, you have to have these things set up, and you also have to have keyless entry to this particular grid set up on your machine, so that we can send instructions to the daemon to do your processing for you. For this, I'm going to set it back to do it locally. And the next one is parse the blasts, and this is where your length cutoffs are applied, or your cutoffs are applied. And then we have a nucleotide RNA. We also put tRNA scan in there. That's already there. And so run your nucleotide sequences through that. Annotate is where it puts it all together and starts processing a bunch of annotation tables for you. It processes all your annotations into pathologic input as what's called PF file, and it prepares all your annotations and sequences as a GenBank file. Prepares sequence files for GenBank submission. There's actually a lot more to a GenBank submission than the sequence file, but the sequence file is definitely a big step in the right direction. Create report files generates a bunch of annotation output, annotation reports. ML tree map is another taxonomy method. You can go read the paper about it, but I'm not going to go in too much detail. It's quite computationally intensive, so I've skipped it by default. And then finally, it says pathologic, and that means let's create a PGDB from my pathologic input. So each one of these you can set to yes, skip, redo, stop, and with the case of blasting RefDBs, you can put this to grid. But as I said, we're gonna, when we do this example, we're gonna do a simple run, not a complicated one. All right, so that gives you a rundown of the template parameter file and all this and how it sort of like encapsulates one sort of run of MetaPathways and what it's going to do. And in the next video, we're going to actually run a sequence through and make sure that everything is everything's set up properly.